So hi, thanks so much for all the talks. It's pretty impressive to hear. I actually changed the slides a little bit I think there's an opportunity here of a lot of interdisciplinary work, and I see people really investigating neutrophils and also chronic diseases, diabetes. So I think there's a lot I'm going to show here that relates to a lot of the talks, also the mitochondrial talk. So it's super exciting to be talking here today with this group. So my lab is at the J. Craig Venter Institute. It's in La Jolla. And we are genomics, most.
Marcelo, you went muted really quick.
Okay, sorry.
There you are.
I'm not sure. So the idea JCVI should do bigger maps, right? So, and when you look at a map, this one has low resolution. It's very hard to advance where you're gonna go because you can't really see. And maps really advance humanity, and understanding where you're gonna go in exact locations can be really helpful. So resolution is important. So you wanna really, head to a area or a region really precisely, and that can be used in really good ways. So in JCVI, there's a lot of involvement in genomics research. Obviously, NIH was also involved, but here I'm just giving examples of maps, right? So sequencing the genome, the science paper with Haemophilus influenza, and then mouse genome and Drosophila, this all helped to create this global impact. And this is just a historical photo many years ago when Craig Venter himself left JCVI and he left NIH and opened Celera. These are all the sequencers that they would try to sequence one genome. And today we have nanopore, which is on the, in your hand. So the evolution continues. You can sequence in maybe 40 hours entire human genome on this sequencer in the nanopore. So, I think science is super interesting if we can think about the fundamental questions and try to develop this map through life. There is that, and there's also microbial research, right? So many years ago, microbiologists found that there's a microbial ecology, and that this ecology is really important. And later on, the term was coined to be a microbiome, which is the seminal metagenomics paper that sequenced, the collection of genes from the microbial ecology impacted the field. And today, there are multiple papers in microbiome. A lot that my lab we are interested in chronic diseases. So it's almost like the combination of these two maps. How can you combine host response with a microbiome response? And could we create a better map in the future? We're not there yet, but that's the goal. One map that's quite shocking is looking at chronic diseases in the US and across the world. So this is not a change of political decisions. This is actually a change in the prevalence of chronic diseases, includes Alzheimer's, obesity, diabetes, and others. And as this is increasing through life and it continues to increase here, you see the bottom portion here, the graph predicting in the future. We think of why, right? The fundamental question is, why are we chronically becoming more inflamed? Why are diseases more prevalent? Obviously, we think about lifestyle, right? Pollution. Those are things that come to mind immediately. Lack of exercise, maybe too much Zoom calls. But the idea here is, why in the molecular level? Could we create a better map? And could we find common markers for diseases? The other problem is that chronic diseases are increasing, but we also have the issue of the lack of target. So for example, if you inhibit inflammation, that could be really detrimental in the long term, right? So we saw the case of cyclooxygenase inhibitors like Vioxx, for example, that created some heart side effects. We know that we cannot take inhibition for inflammation in a chronic manner. There are some protocols that work very well and that can avoid the side effects, but there are many risks of taking non-steroidal anti-inflammatory drugs in the long term. A lot of the pathways that we usually think about are pathways that relates to, molecules that are derived from breaking down the cell membrane, right? Whether this is a microbial damage, whether this is a chemical damage, you have these intermediary molecules like prostaglandins, leukotrienes that will impact inflammation. So a lot of the pathways to block these are to control the enzyme function, whether it's phospholipase, cyclooxygenase, or you can control the cells and activate them to resolve inflammation. And I think Felix has heard, and some others, of this resolution of inflammation, which are lipids that control cells. So in my lab, we focus on human cohorts. We investigate in more details cell biology, and we look for source of inflammation. Obviously, we're very interested in neutrophils because they do a lot of functions, including releasing cytokines, apoptosis, NAT formation, and phagocytosis, and other functions that are not listed here. Neutrophils are quite important because we have 100 billion of those cells produced daily. So if you imagine that, for example, COVID patients are affected in their blood clotting system, you would imagine that neutrophils would be constantly trying to clear that, and maybe NATs are involved in that. So there's a lot of roles for neutrophils in COVID. It's just that we're in the beginning right now. Neutrophils have low RNA, as was mentioned before, and a lot of times people technically cannot study neutrophils because of difficulties of handling and protocols. We have done transcriptomics of neutrophils, and this is a study where we collected whole blood neutrophils, and we did a culture system just to remove monocytes. It's a 98% accuracy to collect just neutrophils. And people, a lot of times when you do PBMC, you don't realize that there's a lot of neutrophils on this layer as well, especially in disease patients. So if you, utilize just the PBMC layer, you are not going to get a lot of neutrophils. So technically, there's an issue with a lot of papers when they talk about neutrophil low heterogeneity. It's because they're collecting from the wrong side. You can collect neutrophils on this side, this layer, and you actually, from disease, you have 10 times more heterogeneity compared to healthy. So in healthy patients, you won't find a lot of neutrophils here, but in some chronic disease patients, you will. And we collect the neutrophils. This is not to go over diabetes, but just to give an example that healthy versus diabetic, these are uncontrolled diabetic patients. You can detect RNA, and you can, in fact, run transcriptomics. Even from single cells, this is more bulk cells. And in this study, we were looking for some genetic defects or transcriptional problems with neutrophils. And we found that diabetics, there's a tendency that most of diabetic genes are underexpressed compared to controls. So a lot of the programs, genetic programs in diabetes, they are oversaturated. So excessive amount of cytokines and excessive amount of T cells. Where we believe that's happening is that in these patients, neutrophils are not transcriptionally active as healthy populations. But there's some genes that's not true. Some genes are inverted, which shows to us that this is something quite specific. There's a global down expression, and there's some specific upregulation. And we're still learning about those genes. For example, in this case, is an adhesion molecule that's quite important for neutrophil function. But when you ask the question, is this reversible? Are these patients, chronically down expressed, but are they reactive to something? So we use these molecules called resolvins, which are resolution mediators. Instead of blocking cyclooxygenase, lipoxygenase, they actually activate neutrophils to do phagocytosis and return to lymphatics. So we added those molecules ex vivo, and you can see that the neutrophils change their expression levels, the diabetics, so they're responsive. So which means these patients are not terminally suppressed. There is a suppression, and there's an immune suppression, these neutrophils, but there's a reversibility component. We look also at the lipids on these diabetic patients, and we show that these lipids are important for resolution of inflammation, like resolvins, lipoxins, murisins. They have a lower expression compared to diabetics. Interesting is that leukotriene B4, which is a major activator, like you saw in Felix graphs, chemotactic molecule, leukotriene B4 is much higher when compared to healthy controls. So there are reasons that the cells are immune suppressed in the long term. So there's my hypothesis that some level of saturation. It could be that there is excessive release of these molecules that then control neutrophil function, but I think there's more to that, and I think there's a microbial and host relationship. This is just an example to show that human and mice are, a lot of people don't like mice studies, and I'm just showing that the cytokine levels of human and mice are quite equivalent. This is DBDB mice and healthy and type 2 diabetics and human and mice. They have pretty similar levels of cytokines. So it's feasible to do mice study. For example, for someone like me that works a lot in the oral, if you add the same bacteria to a wild type mice and normal mice, you have this type of bone resorption. But this is a diabetic mice. So the same bacteria called Porphyromonas gingivalis has been associated to chronic inflammation in the mouth. It was added here, the same was added here. So that's just to show that the same bacteria dealing with the neutrophils were able to preserve some bone here, but in diabetics, you have this excessive osteolytic response. So neutrophils are failing to resolve, or you have excess of some MMPs, which are matrix metalloproteinase, which affects bone osteolysis. These are just trap staining, which are osteoclasts. Osteoclasts are bone resorbing cells, and these cells are more activated in this patient. So what I'm trying to say is that the host also controls the disease aspect, right? So this is the same infection, but different host response affects the tissue. And once you have tissue damage, now you are at risk to develop a disease, because now you have all these molecular events getting to that point. So clinically, it's very important because we may use, for example, these lipids to prevent this, and we use, and it was very efficacious. And it's very important because you can do complex therapeutics and treat disease, or you can go to the molecular level and try to, make sure that these patients in diabetes have similar reactions, they reverse the reaction. So we're trying to develop this molecular map and understand. So in terms of saliva, right? So saliva is a biofluid in the mouth that we are familiar with. And this is sort of like a weird photo to put it like that, but I just want to show that's a whole saliva. That's not a saliva from a specific gland. We have parotid glands, submandibular, sublingual, and other minor glands. But these are neutrophils. So most of salivary cells, immune cells, are neutrophils, and they're live neutrophils. So there are protocols to actually handle those cells, and you can get them live. And you have bacteria, and you have a lot of epithelial cells. So saliva, number one cells are epithelial cells, and number one immune cells are neutrophils. We believe that there's around 1 to 10 million neutrophils circulating in saliva, and some of them egressing and coming back. Just to show, this is a composition, immune composition of whole saliva. So if you just run flow cytometry, you see this. Epithelial cells would be more here, but this is like the immune cells in saliva. And then if you use markers, you can find neutrophils, you can find monocytes, you can find lymphocytes. So saliva becomes a viable fluid for us to investigate functions from the blood as well, right? So we use saliva for diabetic studies, we use saliva for periodontal disease studies. So it becomes a tool. One, because it's non-invasive. Two, it's because it's readily available. So here is just a scanning microscopy of salivary cells. These are neutrophils, these are epithelial cells. And we sort of developed this protocol that you can filter. And it's interesting. When you filter, do serial filtration, you can remove a lot of the epithelial cells, because it's very hard to do any single cell analysis with a lot of epithelial cells in the system. And now you get all this enrichment of neutrophils. And it's funny, we don't get enrichment of the other immune cells. And I don't know why, but it's very replicable. Every time you use this filtration system, you get a huge enrichment of a lot of neutrophils. You can do game cell or H&E staining, all you see is neutrophils. I think one of the reasons that macrophages and some other cells may be more attached to the epithelial cells, and they might be in this model being released or removed by this filtration. So when you look at this enrichment, you don't really see monocytes or a lot of T cells, but you see a lot of neutrophils being enriched in this filtration system. This is just a gating system because we wanted to do single cell. And we did a clustering system invented, it's called DAFI, invented by Max Kian here at JCVI. And we were trying to find heterogeneity of those cells, right? So looking into, these are single cells for neutrophils, looking at different morphology and also different markers to see if they are all the same type of cells or different types of cells. So this is based on flow cytometry and machine learning of neutrophils. So we evaluated the cells in a single cell. We're able to isolate our neutrophils and evaluate it in single cell. And we look, of course, we excluded the other cells because in this case, we only wanted purified neutrophils. The RNA was pretty okay. You know, we could measure, we were doing like cDNAs in these like 304 wells and running each well. We were doing SMART6, so we had to do each well in each plate, the cDNA individually. So we did that. And then we developed this little pipeline for how we were evaluating. Nothing out of proportion. The only difference is that for neutrophils, you do have to have different filtration systems. For example, if you're filtering single cells via eliminating cells with high content of mitochondrial DNA, you will remove most neutrophils. So you have to keep a threshold of mitochondrial DNA there because neutrophils have, you know, high amounts of those genes. So then you can find a map of these cells, these neutrophils, and you can understand that there are some populations. There are different populations of neutrophils in saliva. So blood has neutrophils, lungs have a lot of neutrophils, and a lot of studies just conclude neutrophils have low heterogeneity. But the truth is, it's sort of a false negative in the literature referring to a cell that they were not able to develop a protocol that's appropriate. That's why I was so interested in Felix's presentation, because I think we could use his model to isolate a lot of cells and try to sequence them. So I'll skip this portion, but neutrophils produce these nets that was explained already, and nets are extracellular DNA. And nets, they do this release when they see, for example, a pathogenic oral microbiome. In this case, it's Fusobacteria nucleatum. And that's the release of nets can be pathogenic or could be favorable. So this type of modeling is, here's the neutrophils, in blue are the DNA staining. You start to see a release of these nets. And it's interesting, there's a lot of studies before net formation. Before net forms, there are a lot of like enzymes and dissection of how this process happened. How do the cells produce this release of histones, and what are the MPOs and neutrophil assays doing? But there's not many studies on the structure and molecular composition of the nets after the nets are released. Here is the floating DNA from neutrophils. So it's an open field to understand what's out and about, you know, not just what's producing, but what's being released. So we became very interested, obviously, how the microbiome is influencing that. So one of our postdocs, this is just an example of a study in our lab. We would add like oral bacteria, and we would run, you know, either pangenomics for the different genes in the bacteria, but also proteomics for the host. So when you look at different, and by the way, Fusobacteria nucleatum is present in everybody's mouth today here in this presentation, but there are different types and there are different strains of this bacteria. And this could cause, we did an index for disease, this could cause different types of disease or could influence gnats or neutrophils differently. And here, it's just an example of some pathways that specific microbes could be doing. So there is specificity in the induction of disease, which means there could be gnats that are favorable for healing responses, but there could be gnats that are unfavorable, pathogenic. So we're learning, you know, and we're looking into trapping functions, barrier functions. We're looking into, you know, beneficial functions, immune signaling, but there's also, you know, the vascular damage, right? So gnats, if they're forming the blood vessel, for example, this is like lupus cancer, now COVID, you know, gnats could influence a lot of the blood clots, and this is a knowledge before COVID happened. And neutrophils, they're very important, but what we don't know, it's sort of the molecular composition of this, you know, gnats. We know that there's heterogeneity in gnats, but we don't know exactly the sequence. So we thought to start making these maps, and we looked into neutrophil. This is a neutrophil releasing the gnat. So this is an undifferentiated cell, neutrophil becoming, and then a gnat release cell. We looked into mitochondrial staining, and it's called MitoSox, which evaluates reactive oxygen species. So neutrophils are forming that. They have different levels of mitochondrial, of reactive oxygen species. And it's interesting, there's all this heterogeneity, this low, mid, and high. And depending on the levels, you have a different type of gnats. But what was the question was, okay, what is the actual baseline sequence of gnats? So we looked into gnat release. This is gnats with DNAs, just to show that there's no DNA. And gnats with DNA is on ice, so it is DNA material. Here we did imaging. And then we did the gnat genome, which we call the DTOM. So basically, we have a cell line as a baseline, and then a neutrophil differentiated cell, and the gnats. And each of those bars here it's just one of these. So this is the human genome, and for the chromosomes, and then now we have just each lane is one of those different. So we have a differentially expressed sequence on those gnats. So, which means, say we want to do COVID research, we would like to look at gnat composition. We could use this map to understand how different COVID nets are, or diabetic nets are. And the reason that that's important, that could be that the sequence that's out there in the extracellular milieu, it's very detrimental to the environment. So we don't know that. We need a reference base to start to investigate. So we are looking right now into COVID patients' NAT sequence to understand the difference. Here is just showing this, the previous one was the sort of the entire data. This one is, it's a different threshold. It's mostly the highly expressing NAT data. And this data will be publicly available soon. Just to show to one of the presentations about mitochondria, we had a lot of mitochondrial DNA in NATs. There is most enrichment. If you compare the neutrophils and undifferentiated, there's a lot of enrichment of mitochondrial reads. And we did the proteomics analysis because we have the genome, and now we want to see there's a lot of molecules trapped into that sort of matrix that's being released, right? And we looked into NATs. These are different NAT samples. We sequenced multiple NAT samples. This is an example of 3. And in 3 isolated NAT samples, we had this common amount of proteins. Obviously, there is some variation, but we can see common markers on the NAT composition. For example, the histones that was known before because people do staining. There's also NPO, and there's Elaine. So there's these common markers were there, which made us happy and confident to see that our global maps of NATs, they are reliable. And we hope people will use this as a reference for their own studies. So I spoke today about neutrophils. They're key cells to investigate inflammation. Saliva is an excellent fluid for host microbial studies. We did single cell analysis of saliva. In that case, it was healthy population. We found 8 new populations of neutrophils. We are interested in the composition of NAT. So we did this global map for Genome, genomic and proteomic. And I know that that's very descriptive research, but quite important for where we are right now to start comparing health versus disease. And obviously, maps need to become more specific if we want to predict and monitor lively. And saliva is a great fluid for that type of monitoring. And we continue to do our projects of saliva, here epithelial cells with neutrophils. And here are the, isolation, and we see a lot of the neutrophils by filtration, and we can still see the bacteria around. So it's a great model that we're developing for host microbial interactions. So this is a collaboration work that I did recently with NIH and UNC. And I had my database for oral neutrophils for single cell. They had their database for gingival tissues or oral mucosal tissues. So we were interested in host factors for, related to COVID. So this was a drawing done. We did first by hand, but then someone in NIH did. It's so cool, where we can show all the various tissues in the mouth, right? So a lot of the tissues in the mouth are derived from ectomesenchyme. So there's like real hard tissue, the hardest tissue in the body, enamel, but you have also bone. And then you have oral mucosa, which is highly vascularized. It's the beginning of the digestive system. It's also the beginning of respiratory system. So there's a lot going on in this realm. And there are different tissues. So gingival tissue, you have ducts for salivary, you have different formats of the epithelium tissue. There's also, buccal mucosal, specialized areas like the tongue with neuroreceptors for taste. So we wanted to dissect and start understanding the host factors. Interestingly, that neutrophils in the mouth do not have the direct host markers for COVID, but they have other markers. There are multiple markers, and this was actually published a month ago in Nature Medicine. It was one of the first papers that I ever published that was mentioned in CNN. It's funny to see their headlines. Basically, we use multiple biopsies, mostly from NIH groups and from UNC, and mine was helped on the side to confirm, because we realized neutrophils were not the key host cell marker directly for COVID. In fact, there are multiple new cells in those tissues. So there are cells that are derived from the immune system, like dendritic cells, macrophage, mast cells. But it's also a lot of epithelial cells, and those cells are the most important for host markers. Do we have a problem with time? Okay. Anyway, I'm pretty much done. So just this is the last slide. And to show that, take a look at this paper to show that the oral mucosa, salivary glands, and oral neutrophils and tissues across the body, they have their specific expression of these host markers. And we are currently using proteomics to evaluate cells, neutrophils and saliva from active and convalescent patients. We're not there yet to explain to you what's happening, but we know that we're having a lot of dysfunction with blood coagulation. So with that, I would like to acknowledge people that helped me, including the funding. And it's always good to think about neutrophils as various forms and shapes and functions in the body 24/7. So that matters for those cells. Thank you.